Hello guys, I am Shubham from ShubhVed Tutorials. This video is the part 2 of DBT JRF 2025 PYQ series. So before we start, subscribe to our channel and press the bell icon to get notified whenever new results. This is absolutely free from your side, but it will help us grow. First question, if the pH of an amino acid solution is below its pKa, it would exist as an okay. अमाइनो एसिड हमने देखा है, यू कैन फाइंड इन द बायोकेमिस्ट्री प्लेलिस्ट. So pH is less than pKa. Okay, so pH is basically less, means H plus ions will be more. So protonated form exists, right? Protonated form predominates. Okay, protonated form predominates means what? Cation. Got it? Next question. Transmission electron microscopy involves several steps for sample preparation before visualization. Okay, sample preparation करना पड़ता है. Then last is the visualization. Arrange the following steps in correct order. Okay, of sample preparation. Order we have to see now. So this 5 points A, B, C, D, E. Okay, now इसमें जो first है sectioning by microtome. Okay, then embedding of sample by methacrylate, then staining of sample by heavy metal, then is the dehydration of sample by ethanol water mixture, and then fixation by formaldehyde. Okay, so the staining जो है, staining will be last one. Okay, so staining हमें last चाहिए. C, you will find in option 2 and 3. Then 1 and 4 will get eliminated. Okay, इसमें C1 में तो C first दिया है, that is not possible. Okay, staining will be the last because staining के बाद visualization, right now. And when we consider the option 2 and 3, then BAC common है. Okay, what is C के previous A है, that is sectioning. Okay, sectioning and then embedding, right? तो now starting में क्या चाहिए, DE चाहिए या ED चाहिए? D is dehydration and E is fixation. So first fixation is important. First step is fixation, right? Second, dehydration. Okay, then third is embedding. Then fourth is sectioning. And then fifth, staining. Right? So E, D, B, A, C. Option 3 is the correct answer. Next one, the phi and psi angles of which of the following amino acid homopolymers will occur in all 4 quadrants? See, all 4 quadrants में occur होगा of its Ramachandran plot. Phi and psi angles of which amino acid? Glycine. Right? Easy है. Glycine occurs in all 4 quadrants. Next, in an ultracentrifugation experiment, 2 globular protein X and Y sediment with S value, that is Svedberg unit. Okay, 1.8 and 5.7 respectively. Which of the following statement is true? Okay, X and Y two proteins. X की वैल्यू है 1.8 Swedberg and Y 5.7 Swedberg. तो फर्स्ट इसका होगा ना, and then इसका. Okay, so which is true now? Molecular weight of protein X is 3 times that of protein Y. How? So this is a 1.8. Then 3 times how? Means it is having more weight. It is saying no. Y का weight ज़्यादा है. It will get sedimented first. Molecular weight of protein Y, protein X is 6 times that of protein Y. No. Molecular weight of protein X is lower than protein Y. Yes. Very easy. You can solve. Right. Obviously, जो protein X है lower है than the Y. Right. Molecular weight of protein has no role. Rubbish. In the rate of sedimentation. Right. Sedimentation is having relation to molecular weight. So this is not relevant. The first committed step of the glycolytic pathway is catalyzed by the enzyme. Okay, first committed step, don't confuse with hexokinase. Wrong. Hexokinase is the first step जिसमें ग्लूकोज 6 फॉस्फेट फॉर्म होता है, राइट. फर्स्ट प्रोडक्ट ऑफ glycolysis. Next is phosphohexose isomerase. No. Phosphofructokinase 1. Yes. Pyruvate kinase. No. So option 3 आंसर होगा. Why? PFK1. Okay. So this produces fructose 1,6 bisphosphate. And this fructose 1,6 bisphosphate is actually specific for glycolysis. मतलब यहाँ से सिर्फ glycolysis कंटिन्यू होगा. So this is a committed step, right? Glucose 6 phosphate, the first product है of hexokinase. This can go into glycogen synthesis, right? Glycogen synthesis में उसका रोल है, as well as pentose phosphate pathway में भी रोल है. So this is not only for glycolysis. Apart from glycolysis, it has role in other cycles also. But this fructose 1,6 bisphosphate specific for glycolysis. Okay, so this enzyme PFK catalyzes the first committed step of the glycolysis. Which of the following condition will lead to the activation of gluconeogenesis pathway in the liver? Okay, gluconeogenesis means synthesis of glucose. High insulin, no. Insulin glucose लो करेगा, कम करेगा. So not synthesis, right? High blood glucose, again, already blood glucose level is high. Then why we need to synthesis glucose? No. High ATP and citrate levels. This is inhibitors of glycolysis. Okay, glycolysis को inhibit करेगा, मतलब glucose breakdown नहीं होगा, but this will not synthesize glucose. Okay, no. High glucagon levels, yes. तो ऑप्शन 4 आंसर होगा. ये हार्मोनल रोल इंपॉर्टेंट है. इंसुलिन ग्लूकोज कम करता है, राइट, एंड ग्लूकागॉन ग्लूकोज को इंक्रीज करेगा. So basically this will activate the gluconeogenesis pathway. Okay, you can get confused between option 3 and 4 because if the third option says high ATP and citrate levels, means this will inhibit the glycolysis. Right, glycolysis inhibit hoga, but that will not stimulate the synthesis of glucose. Okay, hormonal role is important. That's why option 4 is correct. Next, different spectroscopy methods are used for estimation of protein secondary structure, okay, and the dynamics. From the following, select the most appropriate method used for estimation of secondary structure of the protein. Okay, just remember this. Secondary structure determination के लिए 3D spectroscopy important है, but this is present in all the options. Okay, you will see here far UV CD. Then in option 2 it is both far UV and near UV. In option 3 it is UV CD spectroscopy and FIDR spectroscopy. And in 4 Fourier transform infrared FIDR spectroscopy only. So these are not the options 1, 2 and 4. 3 is correct because both of these methods are used for secondary structure determination of proteins. Okay, just remember this because technical है 3D spectroscopy and FITR spectroscopy is used for protein secondary structure determination as well as the dynamics. Next, the following gel pattern was obtained when a DNA molecule was sequenced during the Sanger sequencing method. Okay, which of the following is correct sequence of the template DNA? Right, template DNA का sequence हमें identify करना है. So this is the direction of migration they have shown arrow. But when we have to see the sequence of a synthesizing strand, new strand, then we have to go upwards, opposite direction. Just remember this thing. Okay, sequence of new strand. Got it? So what will be that? G A A from down. C T T G C T. Right? This is 5 prime and this is 3 prime. Now what? Complementary strand. Because we have to see the template strand. Right? But don't mark this. Right? इसको mark नहीं करना है. This is a new strand sequence. अब इसका complementary होगा 3 prime. That is A G C A A G T T C. Right? So you will see 5 prime A G C A option 1 correct है. Okay, this is how you will solve this question. Next, which of the following statements is true about competitive enzyme inhibition? Enzymes also we have discussed. Watch that video to understand this concept properly. Right? So I will answer this directly. The first option is correct. It is a reversible inhibition and Vmax of the enzyme is unchanged. Okay, rest other options are wrong. Direct question hai. If you know the concept, you can answer this. Last one. Which of the following bonds joins monomer subunits in biopolymers? Okay, see biopolymers means what? All the biomolecules comes in this, like amino acid, sorry, not amino acid as such, proteins. Okay, proteins in which amino acids are joined by peptide bond. Then nucleic acids, DNA, RNA, nucleotides joined by phosphodiester bond. Then carbohydrates, sugar chains linked with each other, glycosidic bond. Okay, like this. So covalent bonds is answer. Right now, covalent bond is important for the formation of biomolecules from monomer subunits. Right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel and stay tuned for the upcoming videos. Thank you.